I could maybe cut out the iteration. I'm going to try anyway, because I need step function force, and then I need one that slopes a ramp. And I've seen somewhere someone tried a triangular function and then a sinusoidal function. I could try that, but really I'm interested in the slope, in having a mild slope, a steeper slope, and then a step function.
How do you control the step size?
Control the step size?
Yeah.
I just want it to be a step function.
Yeah, one way is to go for more computing time in smaller steps.
Oh, well, it already does that. If you make it between this value and that value, if they're close together, it'll do small steps. And if they're far apart, then it'll do bigger steps to get there. Something like that. I don't want it to do steps. I just want it to do one step so I can get the rate of change. Like I'm looking for rate of applied force because that's very important in tissues. So I need to do an experiment that way.
Okay, can you visualize after a single step? Even though it goes on and does more steps?
I've got it so that it shows the process. Yeah, I've got it so I can see it happening. I don't know how to record that yet.
Take a movie.
It's really strange. It goes down, and then the middle part that I've got, the trijunction, pancakes, and then it flips all over the place, and then and it finally settles.
There you go. You should take movies of some of these art structures when it's windy.
Pardon?
The art, the Pennsegler, the art. You should take pictures of it when it's windy.
Oh yeah. Well, I should build one and then put it out in the wind. Yeah.
Yeah.
And also, if I put water into the model, it slows the oscillations, or at least somebody's already done an experiment like that, and it slows the oscillations if you have water mixed in.
Oh.
Okay, so why don't we get started? So it looks like, yeah.
No, I can't find anything.
It's okay.
Okay. Is anybody who's on using Mac?
I mean, besides what I'm using right now.
What are you trying to do?
I'm trying. I get a message from Jitsi that I don't have permission to use my camera.
Oh yes, but yes, and it's not giving you a dialogue to
Nope.
accept those.
Nope.
Yeah. Sorry, my wife's laptop isn't here, so I'm
on the Lua system.
Sure. I would just need that just to remind myself of what particular path to get to where you need to accept those. Yeah.
Let's see. I'll share my screen. Let's see. Open.
It would be in settings.
Share.
Obviously.
Come on.
Sorry, I'm on my phone too.
Hello.
Right.
Yeah, I had the same problem last week.
Why don't we get started?
Okay, go ahead.
All right. So hello, everyone. Hello, Pakhi and Susan and Morgan, Dick, Jess. Yeah, we didn't have a meeting last week. I was actually at a conference or a workshop, and I'll talk about that in a little bit. Also talk about our community period for Google Summer of Code, and maybe some other things as well. So I don't know if anyone had any updates before we get started in terms of projects or anything else that they're doing.
There was an interesting journal club Saturday afternoon, at least my time, on the organoid intelligence paper that was a multiple lab kind of review paper, really. Well, it was kind of a position paper with a lot of review references, covering, very much the direction of, well, as they said, the one reference that they felt was the closest to what they were kind of proposing in this organoid intelligence framework was the Kagan et al. Paper that is colloquially known as dish brain. So that was, so, it's interesting. Like, I think we were going back and forth in terms of, whether that's a 2D organoid. I would disagree with that, or anyway. But that was dish brain.
Yeah.
And paper where they had a stem cells that were definitely grown to play, and then together with a microelectrode array, given kind of bidirectional communication for playing Pong, and so the idea being that this is where the biology is used as an actual processor, or as a control system. And it was nice. It was two undergrad students from UCSC did the presentation, or presented this review paper, and they are also in David Hostler's class, the Mathematics of the Mind, that I've talked about, and I'm hoping to get they said he plans to release the notebooks as part of like an interactive textbook for this, and so it was nice to see that get a bit of a perspective from these undergrads that were actually I mean they felt a little bad about officially calling themselves brain engineers, because I think that was the more the grad school students, but definitely involved in the kind of brain engineer group that is there at UCSC. And this was hosted by Cortical Labs, so like an actual employee of Cortical Labs. So this was on Discord. So his Discord handle is galactic arachnid, and I only know that his first name is Peter, and he identified as a Cortical Labs employee, but seemed was quite adamant that he's not part of the research group and didn't have any particular insights. Although again, it was interesting to get his perspective on the commercial pressures that they are under, and how that and how that is somewhat limiting in terms of like what approaches that they're taking. And in terms of kind of short-term demonstration. Anyway, it was great. It was nice to be involved, and it was because these students being in Santa Cruz are quite close to San Francisco, so that was nice. And this is coming right on the back of Synbio Beta, where we got to talk about some of the potential, just the intersection there, as well as some of the companies that were there that do this kind of intersection of synthetic bio and stem cell research. Because all the groups that are engaging in this, and there was probably a good 12 groups represented on this organoid intelligence paper, including UCSD. Thomas Harding is the last author, so representing Hopkins, but again, lots of other groups represented. Check just say that. That was really interesting.
Yeah, it's great. So we talked a little bit about that on Saturday, but it's nice to have that update, talking about what they're doing. So what are they doing with organoids in terms of, when people say, organoid intelligence,
right.
Could you tell the group kind of what the implications of that are?
Yeah, so, I said that, despite all the, one of the undergrads had said, that he felt like some of the numbers that they threw out in terms of energy efficiency and like total storage capacity of, kind of like 1 millimeter cubic of neural tissue, seems somewhat arbitrary and perhaps even a little exaggerated. And I did say, it struck me that for an organoid intelligence paper, they had so few, like what I would call neuromorphic computing expertise represented, and certainly all of these institutions have very active neuromorphic computing research groups. So it would have been, for such a still, a multidisciplinary set of people and research labs, like that aspect was kind of missing, and I felt like some of the needed kind of bridge expertise just wasn't there to kind of even talk about it sincerely. Certainly the arguments that they gave are the kind of standard neuromorphic arguments, right? That chip energy use and serial serial execution are the current paradigm and its unsustainable limitations at scale, right? And just that, and that organoid. That was the funny thing, is that they were kind of using neuromorphics arguments and numbers, but with no clear, we can barely, it's one thing for Intel's group to throw around these numbers, right, because they legitimately have like neural processing units that, that can, you can actually get quantitative answers from, but do have remarkably low power requirements in comparison, et cetera, right? So there was a lot of wish fulfillment, or wishing that we could achieve neuromorphic numbers.
Yeah.
but very little in terms of kind of proper breakdown of what did Pong achieve, and and I felt that even their use of the dish brain paper was very this is our best example of what we're talking about, and yet no real breakdown of how easy it was to achieve that with just neurons in a dish. Like even the dish brain part of it seemed almost unnecessary, as like Max Hodak and others have stated privately. Like lots of grad students have trained neurons to play Pong.
Well, yeah, but that's a pretty easy control problem.
Yeah. I mean, to the point. Yeah, which is like, is this? And it kind of reminded me of the criticism of the Indiana group that was doing what was it? It's like voice identification, I think, with the Organoid.
Yeah.
Like, the paper itself was nice from a biology, electrical engineering standpoint, and that was the other issue, where it was just like, why didn't they have anybody whose expertise is kind of brain implants, right, in terms of dealing with the material science interactions with the cellular, cell culture.
Yeah.
In terms of what's feasible, especially for long term. Like, it's one thing to say you're growing these. Again, if you're going to make a supercomputer out of them, right? Like you're using, I would assume you're using the same organoids over time. Like what's going to happen with a lot of long-term electrical stimulation of those organoids, and anyway, lots of holes in it. It certainly wasn't fair to put these undergrads under the gun in terms of trying to explain them. But I thought it was really interesting in terms of getting a group together and their interest. Like I could tell from the background that there was a lot of actual syn bio people there.
Okay.
That was interesting, and that we're kind of looking at it as is this something that my lab would be well positioned to get involved with? That was kind of encouraging too.
Yeah.
And a lot of work still to be done on that in this front. I mean, as the neuromorphic. Oh, and the other thing, saying that I pointed out the fact that open neuromorphics kind of principal is a professor there at UCSC, and maybe we could use this as an opportunity to kind of have, as the next journal club, do something with open neuromorphic. So that was cool too.
Yeah, sounds great. A lot of interesting stuff going on in that space. And, it's kind of interesting how, we talk about, sort of biology and how things are sort of organized and regulated. And then you have computing, and some of the earliest computing work was on kind of taking biological analogs, certainly some of the earliest neural networks papers were about biological analogs, and then now people are kind of coming back to that as a means to see what it can do. So.
Yeah. And I know this is DevoWorm and not biocomputing.
Yeah.
But I'd love to follow up on the biocomputing paper that you covered, I think, a couple weeks ago at the end of Saturday's meeting, and just talking a little bit about that and what I talked about on Slack, which is artificial molecular machines being a generalization of neural nets, like you're saying, like artificial neural nets came from these these simplified mathematical representations. They're simplified mathematical representations of cellular machinery that can also do computation and learning. And that's like as valid and potentially more so for kind of cellular computing with organoids. That was another interesting track from just a few groups at the SynBio beta meeting last week. So what was it called? It was like Alarus Bio. Was a former MIT grad student that gave me some good references on work that his labs there were doing before he started doing this synthetic bio company. Anyway, I'd love to follow up on that.
That sounds good. Thanks for that. I don't remember what that paper was. Certainly I'll have to maybe look it up. But these kind of mathematical representations of biology are kind of also very interesting because
it was like maybe with like certain logic components that kind of biocomputing. It seemed very similar to me as like bios like logic circuits. But anyway, thank you.
Oh yeah, no problem. Yeah, so that's good. So looks like Pakhi is here, and then Mehul was here, but he had to leave. So and then Dick said he found his permissions on Mac OS, system settings, privacy. Yeah, it's always a security issue. Sometimes it blocks the camera. So if you're using Mac OS, you have problems in Jitsi. It's a security thing on Mac OS. So I don't know. You apparently have to give permissions for every app that you have. So, go ahead.
As it turned out to be the browser.
Oh, okay, yeah.
Not just.
Oh, well, yeah, you have to give permissions in the browser, but yeah. So that's good. It looks like Mehul had to leave, but I'll go over some things for Google Summer of Code, our community period. So we're continuing with our community period. And we started at the beginning of the month. So hopefully Mehul and Pakhi have been going through some of the resources I provided, when they were accepted to the program. But I wanted to talk about today some open data sources that we have in the group and how those could be utilized perhaps during the course of your experience this summer. So let's take a look. Let me share my screen. And so, your problems, you'll be working on programming problems, but you'll always need some sort of benchmark data. And I think some of you, during the application period, asked for data, and we have different types of data that we can go to. I think during the application period, we talked about the cell tracking challenge data sets. So there's a website for the cell tracking challenge, and those are nice validated data sets that give you some benchmarks for different algorithms. They've already been benchmarked by different research groups. The problem with those data, or the advantage of those data, is that they're benchmarked. The problem is they're not necessarily specific to what we're trying to do, let's say like C. elegans or development. So there's a range of data sets there that have, they're good for doing machine learning and other things like that, but they have some limitations as well. So we have some resources, fortunately, on the types of data sets we're interested in, and I'll go through those now. So one of the long-standing resources that we have is DevoZoo. This is, we haven't really updated this in a while. I'd like to come back to this and build this out a little bit more. But basically what it does is it offers different types of data that we have in the group. They're pointers to different data sets that we have access to. So some of these are numeric data sets that we have in XLS files or CSV files, things that are tabular. and they're not image data, but they've already been extracted from image data, and they provide numbers and labels. And so we have things for the C. elegans lineage tree. We have things for cell birth and death timing. We have things for the differentiation tree. We have data for cell position, and then some data for gene expression, although this is very limited, and I'd like to be able to add more on gene expression. Unfortunately, there isn't a really good data set that gives gene expression on a cell-by-cell basis for C. elegans, but that's something that, we always have access to different atlases and things like that. So the next thing is we've had these collections of microscopy movies that we've collected over the years. We have like 4 basic data sets that we've worked from in the past. We have this multi-view SPIM data set of C. elegans development, where you have this embryo that's expanding in terms of dividing cells. So you go from a 2-cell state to like a 256-cell state, and you can track the cells dividing, and it gives you pretty good resolution. So you can take that video, unfold it. Some of these already have labels, or they have markers, molecular markers that you can track. There's also some other data sets that are maybe a little bit less good in terms of resolution, but they offer good training sets for you to use, so you can look at the time series of the embryo. We also have data from C-squirt or ascidians. This is embryo data. This is early ceonantestinalis embryogenesis. This is actually numeric data. And then we have microscopy movies that are, track this type of embryogenesis. So this is the C-squirt, which is a different organism than C. elegans. And this might be useful for benchmarking, different, we just usually focus on C. elegans embryogenesis in these projects, but we can also use other organisms to get a sense of some of the things going on during embryogenesis, some of the cell division events, and how those can be kind of detected as features. And then we have the fruit fly data set. Again, these are, I think these are like various, so the imaginal disk is a drawing that you can look at, and you can use it as a way to train your models. We also have some data on fused cell embryogenesis, and I can't remember exactly what those data entail, but we have data from Drosophila. Again, this is just looking at these processes of cell division and differentiation, and it gives you some idea, it would allow you to train a model on these kinds of, processes as they're unfolding. We have zebrafish, we have mouse, spider, and then we have these Bacteria, which are single cell morphogenesis models. So, we're trying to get, we have all these different data sets, and the idea is to get a good sense of, how to characterize these different processes, how to extract features from the process, and then put together some sort of, put it into an analysis or put it into a model and things like that. So that's DevoZoo, and I need to clean that up some. But, it's a constantly evolving thing. We get access to data sets, and, those are, you can use those as you want, put those together in different ways. We have this long-standing repository on GitHub for DevoWorm. This is where we have different data sets. Again, this is a lot of the data sets here are linked through DevoZoo, but we have additional data here on lineage tree information. There are some other things that we've done with differentiation trees, cell birth and death timing in C. elegans. There's raw nuclei data for C. elegans and so forth. So we have a combination of numeric data and microscopy data where you can take the microscopy image apart and have an image for each time point, and then you can, stack those together and say, this is a process that unfolds over time. We have an image here where we have a single nucleus or a single centroid, and then that divides into 2 centroids, and you can track things in that way. And the cell tracking data sets that exist, and I'll get to that next, those are where you have, they'll take high resolution microscopy images, and then they'll track the nuclei or the centroids that they can extract from those data. They do this computationally, and they actually have cell tracking data sets where they can track those in terms of their 3D position. So you have the stack of microscopy images, you have this time series of those stacks, and you can put together three-dimensional volumes that have these centroids in them, and you can track their evolution. So if they divide, if they create more cells, you can get all that information. So when we do things with development, we're trying to get to that point where we have some volume, which is the embryo. We have these centroids in the volume, which represent the cells, and those centroids are going to divide and migrate around the embryo volume. And so being able to track those is important, but also being able to detect different types of patterns in that. And so, whatever the features are there, they can be extracted by the algorithms. Now,
Bradley,
yeah.
A question about that. Does that, when you get the 3D coordinates of the cells, can you identify cell divisions, individual cell divisions?
You can, because the cells in C. elegans are labeled with names of the cells, so we know their general location. And so a lot of the cell tracking data that exists for C. elegans will have labels on the cells, and you can easily label them by knowing their relative position. And so, we can track like divisions, and we know what the cells will become. You can also track divisions through like looking at like a centroid and then having two daughters come off of that.
Okay, in Drosophila, the work by Virginia, what was her name? I forgot her offhand. Okay, at any rate, she discovered that mitoses occurred in waves. Is there any software to detect waves from the 3D data?
Well, we don't necessarily. Well, that's one of the things we've been trying to do with DevoLearn is to detect higher level features. So the higher level features are going to be things that you can detect if we also have membrane data. So sometimes if we have the right data set, people have stained the membrane with markers. Those markers could be like fluorescence or something else, and so the algorithm can pick those contours up, and then you can actually ask questions like, how does the cell change shape? How does the does the centroid split? So it means that's representative of a cell division, and then how do the cells act collectively? And so you can kind of extract maybe higher level features like waves or other types of migration patterns, or there are other kinds of movements, collective movements in development that we might want to be able to characterize.
Has that been done for C. elegans?
I'm not sure people have done that. People have tracked cells quite well, but I don't think people have extracted higher level features like that. That's something
that might be a worthwhile project in itself.
Yeah.
To see what kind of coordinated behavior occurs between cells.
Yeah.
Especially long range in terms of the whole embryo.
Yeah. And, especially for the graph neural networks projects, we're trying to build these graphs. And, the graphs, of course, are not just static. They're adding nodes. They're changing their topology. So if you have a graph that's random, at some point it might not be random. So like a random graph is just where things are connected, in no particular order or in any sort of like hierarchy or anything like that. Eventually you will end up maybe with some sort of structure like a small world network or a scale-free network where the number of connections per node is variable in a predictable way. But that's what happens in the connectome, for example, in C. elegans. So those are things we might want to detect, and those would be signatures of things like, waves moving across the network or organization of phenotype where you're getting differentiation.
Has anyone made a movie of the changing of the network?
Not in terms of a network. People have some of the movies, they show kind of like, cells kind of dividing and the embryo changing shape, and it's hard to detect any patterns within that because, you can look at it and see and say, oh yeah, that's a wave or something, but it's hard to actually put like a mask on it and say, these are the cells that are involved. That's kind of the thing we're trying to do there.
Well, what are these networks of?
Well, usually an embryo network, say, is like cells that are adjacent to one another. We use some criterion to measure like the...
Okay, so the cells would be the nodes and the edges would be the distance between them?
They could be the distance, or they could be like some signaling mechanism like cell-to-cell communication or something like that.
Okay, now if you did that versus time, do you see anything?
Well, we didn't really run that analysis on these networks at snapshots in time and then comparing them. So that's yeah.
Again,
I can only say it. What was that?
Might be worthwhile doing it.
Yeah. Yeah. That I can only say it from my kind of like intuition. I haven't actually produced the graph where there are different network measures. Last summer, Himanshu worked on some things with topological data analysis, and that was kind of where that was going in a way, because you can take like a graph, or you can take like a topology of a graph, and you can look at how you can summarize it with a single parameter, and you can look at it how over time it changes. And so this is like, we didn't really get very far on that, but like that's the idea. You'd have like one or two parameters representing shape, and you would say, there's some shape that we're interested in the data in the network, and how is it changing over time? Is it changing? Is it like you have like a ring, say, is it narrowing down? Is it expanding out? Is it like becoming oblong? Things like that. And then you could measure that over time. So you can measure different aspects of the topology. So that's kind of one way maybe to do that. But again, we don't have that graph that actually shows that very well. It's kind of a reach goal at this point.
Well, in a way you do, because if you have the 3D coordinates, you know which cells were derived from which ones. You can make a graph of that. And in a sense, the graph is three-dimensional and changing with time. It would show you where the cells are versus time, and where so to speak, where the action is.
Yeah.
Okay. So it might be worthwhile making a graph of the 3D positions and just making a movie of that to see what you get.
Yeah. There are a lot of things we can do here. We'll probably get some really interesting data from the summer, so we'll see what we can get there too. But yeah, just kind of coming up with some of these time series analyses would be interesting.
Yeah.
So that's basically what we're trying to do with respect to a lot of the data sets. But then there's also this resource, and this is the systems, the SSBD database, System Science of Biological Dynamics database. And this has access to a lot of different developmental specific data sets. So this is, C. elegans data is here, but they also have data from zebrafish, from E. coli, from Drosophila. And so, these are good quality data sets. These are published data sets with metadata. So you have this, process of development where someone's taken embryos, they put them under a high density or high resolution microscope. They've gotten, information for very short time periods. So, you have this sort of almost continuous flow of movement of cells, divisions, migrations, whatever. And then you have tracking data, so it has some labels and some, like, spatial information that you don't have to sort of add. Of course, you can add spatial information. You could say, for example, add a coordinate system to an image like this. But a lot of the cell tracking software will add in a coordinate, and then you can normalize those coordinates. So it makes it a lot easier to kind of track cells relative to one another. So this is a database that gives a lot of these different resources. We have quantitative data of different types. So this is a stub for zebrafish. This has the nuclear positions of zebrafish. This is from Philippe Keller. This is from a paper, science paper in 2008. They published their data set. Now, some of these data sets are quite old. They're over 10 years old, but they're still useful. They're very useful for getting, the information, and that's what we're interested in. Zebrafish, embryogenesis and C. elegans embryogenesis don't change that much over like 10 years. The technologies get better, but the technology, I think, here is fine for what we want to do. So, we have access to this in a zip file or XML. And so you can take these, and it's an exercise in building a small pipeline. So you pick the data that you want, you open it up, you have a program that opens it up, you have a program that validates the data to make sure it's there in the right order, that you know what the variables are, what they mean, and then you put that in, you analyze it, you maybe produce some graphs, or you can train a model and use that.
Okay, has this been done for the cell sorting problem, or organoids?
What do you mean, like?
Cell sorting is where you separate the cells into different tissue types, and then you put them back together. They're initially random, and they sort out, make onion structures.
Right.
Okay, this is the old Treehouse, I think, started it, and Malcolm Steinberg continued this work.
Right.
Okay. So, if there uncertainties about what was happening, so if you could observe it in 3D and track the individual cells, you probably get some interesting information about what they're doing.
Yeah. Yeah, I mean, that would be, another option to work with this. Yeah, there are a lot of things you could do with it, I guess.
And there's all sorts of organoids around, so it should be possible to do with organoids too and see what's going on.
Oh, yeah. Yeah, there are, now, I didn't even mention organoid data sets, but there are open organoid data sets. I know Morgan would probably know.
There are.
Some good. Yeah, well, yeah, there are some. I mean, I don't know. Like, some of them are electrophysiological, because a lot of people are interested in, like, what they're doing that way, but they're also, I think they've done some cell tracking on some organoids. But they definitely have, like, organoids that have been stained, so they have, like, microscopy images of them, things like that. So, so I mean, there are a lot of things we can do with these data aside from, like, training models. We can, look at different tissue types. We can look at different, higher level features. We can do all sorts of things like that. And so the point of this was just to go over kind of some of the resources that we have on these things. And, if you need something, some sort of data set for your GSOC project, we have those available. And we have other types of data that we can find. This isn't the limits of data that's available. So there are all sorts of data sets that are out there. Some are, we want to make sure it's decently validated. In other words, we want to make sure maybe we have labels, or we can label the data if we need to, or that it has, that it's, if we want to take multiple microscopy movies, that we can align them properly. We don't want to just, kind of grab the data. Sometimes, we have to do that. Like when we were doing the digital Bachelararia project, we had all these videos from YouTube. People have taken videos of Bachelararia colonies, and you just extract them, and you kind of take the data and you build a little data set, and it's a pretty simple process that you want to isolate, so it's not a big deal. But it's it's still, you want to be able to align the data properly, and so making sure you can do that is an important thing. So there's a pre-processing step, and I wouldn't spend a lot of time on that, but I would be mindful of it. And then there's the analysis step, getting it, analyzed to a point where we can then make some insights for graphs and other things. Okay, so I think that's enough on that. Now I wanted to talk about what I was doing last week. Well, we didn't have a meeting last week, and that was that I was at a workshop. Okay, yeah, I'll share a link in the Slack to the SSBD dataset. So I was at a conference where we were talking about building minimal cells, and this is interesting because this workshop was put on by a group that had been modeling what they call minimal cells. So there are these cells that are, if you're familiar with the J. Craig Venter Institute, they've been building what they call the minimal bacterial cell. And that bacterial cell is like the minimal cell you can have that's still alive. So it's got just the genes you need for energetics and maintenance, and that's it. Then you can boot up with new genes, for specialized things. But, you're working from a minimal cell. So the upshot of that is that you have a model something that you can model relatively easily. You don't have to worry about a lot of things like, different organelles or things like that. So I'm going to talk about that. I'm going to go through a couple slides and then some resources on this. So this is building minimal cells, and this was this workshop here. This was put on at the University of Illinois. This is called the Advanced Computational Workshop, Simulations and Visualization of a Minimal Bacterial Cell. So these are bacterial cells, not animal cells. I think they've tried to do some work in yeast so far, so they're moving up from bacteria to yeast, but they're not there yet. So their focus of this workshop was on bacterial cells. So they covered a number of topics here. It's all week. So they talked about the minimal cell, which is the JCVI SYN3A, which is the standard workhorse of that. And they had to figure out how to model that. So they have the sort of the biological specimen. Then they have to figure out how to model that cell. So they have to figure out how to model the genome and how to model the proteins. And we're talking about basically a vesicle. So it's not a hugely complex thing, although you'll see that it looks pretty complex. We use a number of computational models and put them together in a pipeline to simulate the cell. So this is kind of what we're doing in OpenWorm with some of the different tools. So we're modeling movement, we're modeling the neurons, we're modeling the connectome, we're modeling the muscle and all that. And so they're doing this with a bacterial cell by putting together different tools and producing a visualized output. So if I go back to the slideshow here, this is JCVI SYN3A. This is a mycoplasma. And so this is actually a species they call Mycoplasma laboratorium. This comes from Mycoplasmic genitalium, which is a mycoplasma with a bigger genome. And what they've done is knocked out the genes that are non-essential. So they do these knockdown experiments, combinatorial knockdown experiments, where they would test each cell, basically test each gene. They remove the gene, and if the cell died, then it was essential. If the cell didn't die, then it was non-essential and it was removed from that genome. So in doing so, they could generate a minimal set of genes that are required to sustain life from the genome. And so, they've done some additional genetic engineering to enhance some of those genes to make sure that the line can be propagated. But they end up with 482 genes and 582,970 base pairs. So if you look at this picture, we see all the constituents of this type of cell. So it's basically a vesicle with a lipoprotein membrane with soluble proteins inside these purple circles, these purple balls. Ribosome, which is this green ball, this bigger green ball. You have these black things that are twisted around here. This is the DNA. So it's a circular genome, but it's kind of packed in there, and it looks like it's twisted around like a coil or a cord of some type. And then you have soluble RNA, which are these yellow like squiggles, and all of that is packed inside of this vesicle. So you have a lot of stuff in there, and but it's not like hugely complex. You can model this using molecular dynamic simulations, and that's what they were talking about in this session. So if you look at the model that they build, this is the computational model here, as opposed to the mycoplasma here. This is where they've modeled all of these components, and they're packing in this inside this vesicle. They have the outside of the vesicle with receptors on the edge, and then inside you have all these things that are packed inside. You have this sphere of a certain volume. You have water. You have the physics of water, and you have all these components like DNA, RNA, proteins. And so we're at a very minimal state, not only in terms of the genome, but in terms of what's inside this vesicle. And so the idea is that you want to model this as a single cell. You can do this in two ways. You can do this using a lattice microbe methodology, which is where you basically have this whole inside, which is the cytoplasm. And you sample the cytoplasm for the different components. You look at the metabolism, and you use a chemical master equation to estimate what the inside of the cell is at any time. And so you can build these models from the sampling, kind of what the constituents are of DNA and RNA and protein, and you can do these different estimates. You can also use something called Martini or Gromax, which produces beads. So you can either have this lattice, which is like a bunch of points, and like this lattice, it's kind of like a triangular mesh or some sort of a rectangular mesh that covers the entirety of the vesicle, and you can actually put things inside inside that mesh, so you can have like a triangular motif, and there are things inside of that, and you model that as a unit of the cell. And you can do that with lattice microbes, or you can do this use this Gromax package where you produce beads, which are these like little volumes of coarse-grained volumes of this space. And inside the volume, it's almost like a voxel in neuroimaging, where you have things inside that volume, different chemical reactions, different atomistic reactions, and that's how you would represent what's going on in the cell. So, they are able to look at DNA replication, transcription and translation, so you can go from DNA to proteins. You can generate trajectories. So what they do is they basically simulate this in these different ways, and then instead of predicting every interaction over like maybe 50 to 100 microseconds, they generate trajectories. So they say, what is the mean behavior of this model, and what does it look like over time? What does the metabolism look like from sort of our initial condition to maybe like cell division? And so that's how they get a sense of what the minimal cell is doing over time. And then the visualization step involves something called VMD, which is visual molecular dynamics, or they actually are using Minecraft, which is a program that creates these virtual worlds that you can interact with in a VR headset. So the lattice microbes methodology is where you have these lattices, and things are represented in these lattices like membranes and ribosomes. You can simulate these on high performance computing nodes. So this is something that requires a lot more than like a laptop. You use like many cores to do this, and you can set up a job, and there's you need a lot of computational power. What you do is you sample the cytoplasm within the minimal cell. You feed it into a chemical master equation. In this case, that's a reaction-diffusion type equation, and you can make predictions about what's going on in the cell at any time. And then, of course, they have different assumptions. So Sometimes you can make the assumption that the cell is well stirred, meaning that the components are just kind of drifting in there at random, or sometimes there's spatial segregation in the minimal cell. So sometimes things are clumped in different places. This is maybe, you can use both assumptions. The homogeneous assumption works most of the time, but when we start getting into cells that exhibit differentiation, we're going to have this problem of heterogeneity. So that adds computational. We need to have better computational resources devoted to that. And this Roberts et al. Paper, yeah.
Yeah, one curious thing about this work was that the homogeneous was good enough in most cases.
Yeah.
Which means that the actual three-dimensional structure inside the cell is not important.
Yeah. Well, yeah.
They're just the concentrations,
right,
in substances.
Right. Yeah, I mean that's one of the things about like these models is like you're making trade-offs. So you're saying that you're making assumptions about sort of the the energetics inside the cell, the metabolism. It's not often in equilibrium. So, but you still make an assumption about the behavior. You don't try to model every interaction. You try to model sort of the averages. And then again, the 3D resolution, you can model it with these beads, but really you're making the assumption that there's no particular structure that you need to replicate. So you can just take like things at random and say, well, everything is basically well mixed. It's fine. And it saves you computational effort, because otherwise what you would have to do is you'd have to model subcomponents, and they'd have to be modeled differently, and that sort of thing. So this Roberts paper kind of goes over what lattice microbes are. It's a stochastic simulation method for modeling this reaction-diffusion mass transfer. It's very much chemistry and physics based modeling. So this GROMACS Martini, GROMACS is a package that allows us to do this modeling where we're producing a trajectory in the computationally cheapest manner possible. So if you have a system, you want to be able to produce a trajectory to say, what is the state of the cell? What is the state of these different components of the cell? And how can we estimate that? We can't really, again, measure every interaction, not least of which because they're not necessarily important to get those precise, but because we can't, we don't have the computational power to do that. So, we try to approximate things in this. We try to approximate things like hexagons and bounding boxes. And they were talking about these different methods like triclinic and rectangular methods, which reminded me of how we think about like cells. We think about trijunctions and quad junctions and cells and how those are important. And that's one of the things that they're doing with modeling. They're trying to model at that level of like what's interacting with what. We have these like kind of volumes that we can, the subvolumes that we can take, and how do they interact with each other. And then this paper here, Pezeshkian et al. 2020, backmapping triangulated surfaces to coarse-grained membrane models. This is where you have these surfaces that are in this triangular mesh, and you're mapping that to sort of a coarse-grained model where you have these smaller volumes that tell you kind of, what are the atomistic interactions in this volume. And, it's just, we're not trying to model every atomistic interaction, not trying to model like water molecules. We just want to know what's going on in this little volume. So, we use force fields, physical force fields, to simulate like things like water, things like interactions of proteins, etc. You'll use repeated motifs, so that cuts down on your computational time. So if you have a bunch of triangles that are doing the same types of things, you might use that as a single unit in your computation. And then we have this coarse graining method, which is where we choose a higher level of analysis. So this is important when we do things like try to abstract things to the phenotype. And this is especially true in like whole organism modeling, where, we say, well, what do we want to model? Do we want to model every molecular interaction? Do we want to model every atomistic interaction? Is it just good enough to just model a cell as a cell and maybe have some features as a cell? Or maybe we don't even care about cells. Maybe we care about different higher level features in the phenotype. What are those doing? So this is a term they use in this area called coarse-graining, which means you take the simplest interactions and you combine those into maybe volumes or motifs, and you kind of model those things, and you get the sense of what they're doing. And so it's a choice. You can model things with high precision, every interaction. Sometimes it doesn't matter, sometimes it does. But, sometimes you just say, well, I want to coarse grain things because I really don't care about these lower level things. And so this is all about modeling and the choices you make during modeling. So they do a number of things when they do this. They build a vesicle, build a membrane, introduce proteins, polymers, and DNA, and then we also introduce some water. So we build a simulation by introducing these different things and then putting them all together. We'll use statistical mechanics ensembles to model the dynamics of chemical species, reactions, and particles. So all those things in the model, in the volume. We'll do things, one of the things they do is they build this pontylized mesh. So we talked about pontylism in the meetings, and this is something that Dick is interested in with respect to medical imaging. But they actually use pontylism in this modeling framework as well. And that's interesting because, and I'll show some stuff on it real quick at the end of this, it really allows you to deal with things like coordinate systems and curvatures. And there's been a lot of interesting work in computer vision on pontylizing things in modeling. So it's an interesting point I picked up, maybe to look into later. There are a lot of open data sets we can use, metabolomics data sets, proteomic data sets, and genomics data sets. So, these are things that are open to people to use. You take some data, you use it to sort of inform your model, you add all your components in, you estimate things from the data, and then you have your simulation. So your simulations, it depends on what you want to do. So in these minimal cells, they simulate things at tens to hundreds of microseconds, and that doesn't seem like a lot, but actually in terms of like the molecular reactions, the scales of those are much smaller. So when you have your time step in your simulation, what are you representing in that time step? Is the reaction of interest or the phenomena of interest sort of, do you coarse grain your model in time well enough to pick those up? So if I'm interested in embryogenesis, I might want to coarse grain or fine grain or coarse grain my model to some time step that allows me to see the relevant dynamics. So if I'm interested just in like protein folding, I want to have a very short set of windows. If I'm interested in maybe like cell division, I might have a longer set of windows, that sort of thing. Or not, cell division is interesting because there are a lot of things happening at very short time scales and then things happening at longer time scales. And then this gets visualized into this kind of a model. And then, so one example problem they gave was of cell division. So in their models here, these are static models in the sense that they're just this model of a cell with stuff in it. But you can actually have models where the cell divides. And so, that this outer membrane needs to stretch out, elongate, and then all this stuff needs to double in number. So all the DNA, all the protein, the components need to double, at least the DNA needs to double. And so the idea here in the simulation is that all of these components need to double in number. But they don't, they need to do this as the vesicle membrane is stretching out. And so as it's expanding, you're eventually doubling the number of the components. The water is expanding everything out, and then eventually you get this division where the membrane of each daughter cell closes, and then you have two cells. And so that's what they were looking at modeling. Now, we have a problem where we need to represent this properly, so sometimes, we need to take these contents and we need to segregate them out to each pole of this elongating membrane. We need to then have something in the middle to pinch off the single cell into two cells. So the vesicle membrane must grow, stretch, and pinch off accordingly. And this all needs to be done with these lattices and beads and things like that. So it's not that we're actually observing this process, it's that we need to model this in the context of our digital model. And so we can simulate different mechanisms like FTSZ, which actually accumulates as a ring in the center of a cell or a vesicle that allows us to have this sort of cell division where this FTSZ ring like contracts and pinches off the membrane. So, you can simulate that process. That's a protein-driven process. You can actually also simulate this segregation of materials, this doubling in number of materials, and this stretching of the vesicle. So all those things we need to model for this process to happen, and they showed how this can be done in their models. So, this is Mycoplasma Laboratorium. This is the Wikipedia page for it, if you're interested. Then, there's some open source software associated with this. So this is the software for the workshop. This is actually a vesicle that's dividing. So this is a vesicle that's stretching out and doubling the amount of DNA in the vesicle. So you can see how this thing stretches out and starts to pinch. They have code here for the DNA model, or this model as well, which is the tutorial. I mean, this is just stuff that you can't really do on your own, but you can run it if you have access to a cluster. You can run it. And then there's some images in here where you can see some examples. There's also this. This is the GrowMax open source repository where they have a number of tutorials, and it kind of goes through how you would build these simulations, the kind of commands you'd use, but also the kind of processes you need to simulate. And then there's this VMD, which is the visualization tutorial. So this is a visualization program you can install. You can install these on your own computer or on a cluster, and you can do really interesting things with them. So I wanted to get back one last thing before we go, which is to talk about this pointillism aspect. So I found a number of papers. So this is the Pezeshkian paper on backmapping triangulated surfaces to coarse-grained membrane models. And in this paper, they kind of talk about this pointillism process. So they talk about backmapping on a lattice by sort of introducing these points. So you can see these points on the triangle. And so you're trying to refine this mesh in terms of its shape and in terms of, what's inside of it. So you're using these points to do that. So they use this pointillism process. They use it to grow vesicles as well. So they're backmapping. What backmapping means is they're refining everything. So they're able to use these beads to refine the mesh and make it more realistic looking. And so this is a screenshot here of pointillism. This is from the method section, where they talk about pointillism extending the upper and lower TS. So in this step, the number of vertices of each TS, upper and lower surfaces here, this is the triangulated surface, will be increased to a larger value than the required number of lipids to fully cover their surfaces. To do this, we first need to make an approximation of the geodesic, which is the curvature, connecting two neighboring vertices. Consider two vertices that are connected by a vector link 1 of size L. The projection of 1 on each vertex plane is denoted here. We require that a geodesic connecting the two vertices satisfy 3 conditions on each vertex, 6 conditions in total. So they go through this, and they're basically using these points to refine the mesh, the triangulation mesh, and the geodesic. So this is actually something that has been done in, we can build algorithms for pointillism, and we can do pointillism paintings in Python. So this can be implemented actually using Pygame as a toolbox. And so we can actually do this. And so pointillism has been well studied in computer science, and it's been used in computer science for a number of applications. This is an example of pointillism just in general, so it's not in a biological simulation, but they've been able to do this simulation where you can generate points in a model, and then of course this paper here, which is a 2013 paper, Eurographic Symposium on Rendering, where they talk about generating pointillism paintings based on Seurat's color composition, where they can get these images that, they use pointillism, they're mimicking different paintings and they're doing this algorithmically. So the point is that you can get these different techniques in quantilism and put them into an algorithm and generate things for different applications. So this is, computer science, hardcore computer science, but it's something that can be used not just in fine arts, but in biological modeling. So that's all I wanted to talk about with respect to that. I thought that was a really interesting workshop, and they have their model of like simulating cells, right? It's not like, that's not what OpenWorm does, and it's not what other groups do. But you're working from this sort of first principles, this ab initio type of approach, where you kind of know the biochemistry and the physics, and so you're working from that, and you're just assuming that you can build a model up from that and build a cell.
Okay, I've actually used, well, I call it pointillism,
Yeah.
sort of tomato thing. But I've used it for my paintings.
Yeah.
Since that's my hobby. And there's also a CompuCell 3D workshop this summer, end of July, I believe.
Yeah.
And I found a YouTube video about one of their sessions, say 2015, 2018, I don't know. So I'll send that to you.
Okay. Yeah, that'd be good. Yeah, so the other thing we can do is compare this approach with CompuCell 3D and how they kind of approach the problem of modeling cells. And of course, they use a different method. They use a kind of a different kind of method where you're modeling cells in this lattice and you're doing physics on the cell. And it's, you're not worrying so much about what's inside the cells. You're modeling the cells themselves. And you do inform what they do. There's, data about what kind of processes inside the cell, like gene expression or something. But it's not like you're not modeling it down to like DNA content and proteins and, getting those things in place. And
and they're not doing tensegrity either, which is very disappointing.
Yeah, I was thinking about tensegrity throughout all this. I'm like, how would you model tensegrity and how? Because it seems like it's right there. I don't know how it's done.
Well, I don't like the way my finite element analysis is doing it, but I'm going to live with it.
Yeah.
For now, try to get it to stop being so automated.
Yeah.
It goes, oh no, it's not settling down. Maybe it's broken. So then it redoes itself and puts another force on it. It's like you're putting pulses on the tensegrity, and of course it wobbles all over the place.
Yeah.
Just, not too coordinated.
Yeah.
I've been having fun with that.
So Dick says, shape of a pixel not important equals an example of pointillism. So what is that? Could you elaborate on that? Or?
Yes, in computer tomography, Alicea Mehul, what she did is you have to project a pixel onto a line. And what she did is allow rotation of the pixel so that it projected as the same width as the pixel, but no matter what the angle was. So the pixel was allowed to rotate.
Yeah.
And this actually improved the computer tomography pictures.
Yeah, good. Yeah.
So it was kind of strange. You start ignoring different features of the pixel, and it gets better.
Yeah.
It's sort of like these retina screens. If you make your pixels on the retina screen small enough, it doesn't matter what shape they are.
Right. Yeah.
Okay.
Yeah, that's the other thing about modeling. Like, we can ignore things, but we have to ignore only the right things, or else we miss.
Yeah, in this case, we kept the density of the pixel, but not.
All right. Well, I think that's it for today. Did anyone have any other things they wanted to talk about before we go? Or?
Yeah, I guess so. Are you getting anywhere with the curl and noise?
Well, a little bit. There's still some stumbling blocks there, I think, but we'll figure that out. Yeah.
Okay.
It's hard to find like existing software though. Well, it's kind of like your packages for like computer graphics, and it's very focused on that. And then like using it for other purposes is less easy to actually get it to work. I don't know if that makes any sense, but
Okay. Yeah. There's Python code for generating Perlin noise.
Right.
Okay. Which we can pick up, but I think were you able to get the parameters out of that paper on restrooms?
Yeah, I'm still working on that.
Okay. Because that's kind of that paper is interesting but poorly written.
Yeah.
Well,
yeah. Okay. All right. Well, thanks for attending today, and talk to everyone next week. Again, we're going to be still in our community period for GSOC, so I don't know. I'll probably come up with some. Maybe we'll go over some of the papers that we've done and talk a little bit about that, so that new people know kind of where we've been and where we're going. So I also have put out a video on the 10 years of DevoWorm. So that's something I talked about in the meeting a couple weeks ago. I actually have a video now on the YouTube channel. It's about 90 minutes long, so it's not trying to torture people, but there is a lot there. And with the YouTube videos, you can actually go through chapter by chapter and find interesting things. So
what is it called?
10 years of DevoWorm.
Oh, okay.
Yeah.
Bradley, one thing about the small cells or the mineral cells, is that they confirm the concept of autocatalysis. And that is, if you throw the right chemicals together, the chemicals organize themselves.
Yeah.
Okay, so autocatalysis, while it hasn't been demonstrated, in the sense it has been demonstrated through the minimal cell simulations.
Yeah.
The other thing is they got about 75% of success. In other words, 15% of the cells didn't work.
Right. Yeah.
Okay, but that's not bad for a start.
Right. Yeah, like a lot of cells, I guess they were saying that their experiments that they do with minimal cells, there's a significant amount of cell death. And one of the challenges is to figure out why they die, because not every cell dies, but when you're at that minimal set, you're not going to have, like, you can survive, but maybe not in every condition. So it's like, why did they?
Yeah.
Okay, thanks for attending. See everyone next week.
Bye.
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 Bye. All right, so one additional thing related to all the stuff we've talked about biological modeling of minimal cells. This is an recent article from Stephen Wolfram on biological evolution and other adaptive processes and using a minimal model to capture the attributes of those systems. So this is from his blog. Basic, as you could guess, he's using some sort of cellular automata. So let's go through the model and then some of the other things in this article. We have the model. Why does biological evolution work? And for that matter, why does machine learning work? Both are examples of adaptive processes that surprise us with what they've managed to achieve. So this is this idea of computational universality, where you can basically take biological systems and machine learning systems and find the fundamental aspects that underlie both as complex systems. So he's going to concentrate on biological evolution, but then this is also relevant to machine learning, and this is going to be the subject of another article. So what's the appropriate minimal model for biology? So we talked about the minimal models that they're working on with mycoplasma, and that's having this vesicle with things in it, with stuff, packed stuff, and it has kinetics and it has metabolic activity, and it divides, and you can simulate what's going on inside the cell and also division of the cell and other things like that. But sometimes we want our minimal models to be even more minimal than that. So this is where we have cellular automata. So cellular automata, of course, is this grid of cells where each grid has certain property, each cell in the grid has certain properties. It can transition between states and so forth. So in this case, he builds a cellular automata, an example using cells with 3 possible colors. The rules are shown on the left here. So you have these transition rules depending on what your neighbors are doing. That influences the state that the focal cell takes on, and then that is applied in parallel so that you get these sort of collective behaviors. So this is the minimal model here. We start with a single red cell, and we see from this seed a structure is grown. So we start up at the top with a single red cell, and we get this structure of red and purple and white cells coming down. And so a structure is grown, which in this case dies out after 51 steps. So at step 51, we end up with a single blue cell, and then it dies. So we end up with a genesis of a cellular structure, the evolution of a cellular structure, and then its death. Now we can model this in cellular automata in this minimal model. In this case, we're modeling cells interacting with each other as a multicellular agglomeration and then dying out. We could also think about this as a bacterial colony. But, this is very abstract. But let's say we start from the trivial or null rule that makes any pattern die out immediately. And we end up adaptively evolving to the rule above. Imagine making a sequence of randomly chosen point mutations. So in this case, we have a one-dimensional array where we make point mutations. And essentially, at any one point in this one-dimensional array, we change the state. So we mutate the state to a new state, and then we evolve the system and see what happens. And we end up adaptively evolving to the rule above. Imagine making a sequence of randomly chosen point mutations, each changing just one outcome in the rule, as in this example here. Then suppose in each step a minimal analog of natural selection occurs. So our minimal analog of natural selection involves accepting any mutation that makes the lifetime longer, although not infinite, or at least the same as before, and we reject any mutation that makes the lifetime shorter or infinite. So we basically select for longevity. And so we accept the mutations that allow for longevity, but reject the mutations that limit the lifetime of the structure. So you can see that, based on the interactions between cells, sometimes if you have these one-dimensional arrays that are kind of laid out like this, you get this, you get states that persist. If you mutate them in different ways, you get states that get overwhelmed. And this is just simply a product of interactions between neighboring cells. In cellular automata, sometimes you get convergence to a single state. Sometimes you get convergence to all zeros or something like that. So this is something that happens in one-dimensional arrays as a consequence of its evolution. So we can actually mutate these rules so that you can avoid this convergence and allow the structure to persist. So it turns out that with this procedure, we can indeed adaptively evolve to the rule above. Here we're showing only waypoints of progressively greater lifetime. So in this example here, we have a single red cell. We end up with this multi-state agglomeration which grows and continues to grow and expand across the grid. And so we can see this at the final step here, where we have this very complex structure. And you can think of a colony of cells having this property. So, you might have a colony that dies off quickly. You might have a colony that undergoes a burst of growth and then dies off. And you can have colonies that persist at very complex levels of significant levels of complexity over time. And so different sequences of random mutations, in other words, different ways of changing these states, give different sequences of rules. But the remarkable fact is that in almost all cases, it's possible to make progress and routinely reach rules that give long-lived patterns, here with lifetimes 107, 162, and 723. So this is, these can persist for quite a while. This can persist up to 700 generations or more with elaborate morphological structure. So here are some examples where you get these cellular automata structures that persist for very long time periods. And this is, in this case, over 700 generations. So this is an interesting thing. I think this is another interesting thing about this is that they talk about making progress. And there's this whole sort of controversial idea in evolution about progress in evolution. And it's tempting to think of progress as like, something that is, intentional. But really, it's just the product of certain conditions. So, in life, we tend to see more complex life as we move, forward in geologic time. So, in the early years of Earth's geology, in the early years of Earth history, we had very simple organisms. And those organisms became progressively more complex. And now we have a diverse tree of life with incredibly complex life. And so there's this, I think Stephen Jay Gould termed it the wall of complexity, where you start out at a minimal state and you evolve through these very complex phenotypes. Indeed, you can see that in the data. But, is this really the imperative of some sort of progress? Or is it just based on the conditions that exist? And so this is arguing sort of that those conditions have to be there to make progress. And he puts it in quotes. And I would definitely put it in quotes because it's not something that we want to confuse with what's actually going on, which is that we give sort of this cell collective the right sets of conditions. And we're modeling those as cellular automata, which in and of itself I think is a little bit abstract for what we want to do, but it gives us interesting results, and that's what's important. Looking at the sequence of pictures above, we see that there are often in effect different mechanisms for producing long lifetimes that emerge in different sequences of rules. So we can produce long lifetimes, we can have different rules for getting there, different ways of getting there. So we have different mechanisms for building complexity, and in fact that's also what we see in the tree of life, is that complexity can happen in a wide variety of branches of the tree of life. But let's drill down and look a little bit in a little more detail what's happening in the simple model we're using. In the three-color nearest neighbor, k equals 3, r equals 1 cellular automata, there are 26 relevant cases in the rule. Point mutations affect a single case, changing it to one of 2 possible alternative outcomes. So that altogether there are 52 possible distinct point mutations that can be made to a given rule. So again, these are just point mutations. This is just changing a single cell. And then when you change a single cell and you keep evolving the system and changing another cell, you end up with an explosion, combinatorial explosion of possible point mutations and diversity in behavioral outcomes. So we see that from this seed, we can mutate in different ways at the beginning of it, and we can end up with all these different behaviors. In quite a few cases, the pattern generated is exactly the same as the one for the original rule. In other cases, it dies out more quickly, or it doesn't die out at all. So we're working on this approach using cellular automata called the EMCA approach, and it's using these one-dimensional automata. And we're finding some similar results. And I find this interesting because not only because he's doing some experiments with minimal sort of cellular systems, but because we've kind of seen similar results in some of the work we're doing. So this is an interesting article. So he goes through a lot of this, and then he talks a little bit about sort of this dimension-reduced rendering of the 27-dimensional rule space. So he sets these rules in a rule space and builds this network of rules. And, basically the idea is your phenotype will sojourn through this space, through these different conditions, And there are periods when there's lots of wandering around going on, with many mutations needed to make progress. And there are other periods when things go much faster and fewer mutations are needed. So we can see from this plot here that we have this number of, generations. We can plot the maximum lifetime achieved so far against the total number of mutational steps made. So obviously, the longer something persists, the more mutations it incurs. And if it incurs mutations and doesn't survive, then it dies. And so sometimes mutations can be good, and sometimes mutations can be deleterious. It just depends on if we pick the right ones. We see plateaus, including an extremely long one, which is here, where no progress is made. And I guess progress meaning persistence through mutation. And I don't really know if that's progress. I guess, the mutations sort of allow the colony to persist and they build upon one another. So in this case, if we have a one-dimensional array where we mutate some site, it has a downstream effect. And so in that sense, these mutations can be cumulative, and the memory of that isn't in the genome, but it's kind of read out in the cellular automata. So it sort of unlocks certain pathways, certain patterns, and then allows those to persist. So this is an interesting paper, and then this is where he kind of gets at some of these. So far we've mostly been talking about one particular random sequence of mutations. But what happens if we look at many possible such sequences? Here's how the longest lifetime, or in effect fitness, increases for 100 different sequences of random mutations. And so this is the thing here where we have, we go out to a certain number of time steps, and we get these mutations. So we get, this is mutations on the y-axis, this is time on the x-axis. And so here we're kind of exploring this space and exploring where we're getting sort of these adaptive processes that emerge where the population doesn't get stuck, it allows it to persist. And then he talks about multi-way graphs, which are these graphs here that show all possible mutation histories and how they're connected to one another. So a couple other really interesting things from this article. It's very long, so I'm not going to get into all the details. But he talks about multi-way graphs and how that sort of shows the connections between different types of rules and things like that. Then he makes the link, of course, to fitness landscapes. So a lot of this work is very reminiscent of what we know from artificial life research. And so it makes sense to look at the fitness landscape to bring that into play. So he does some work on looking at fitness landscapes and what he calls fitness waypoints, which are decisions or decision points within the fitness landscape that allow a population to move in one direction or another. And sort of I don't know if you want to call it a decision point, but it's definitely a point of contingency. In other words, if this is the rule or this is the mutation that's made, the population will go in one way versus another. And then you can reconstruct that and show that sort of contingency as the product of a series of contingencies as sort of the product of evolution. And so, we can also think of this in terms of exhaustive versus adaptive search. So exhaustive search would be where the space was searched randomly or brute force, and we can bring in ideas about adaptive evolution and adaptive search to the space. And so he talks about this a little bit. Then he gets into some of the issues in computation. So he talks, the first thing he talks about is this issue of undecidability. So this is where we talk about these fitness criterion and how patterns either terminate or don't terminate. And then he asks the question, but how can we tell if a pattern is going to terminate? So sometimes patterns live for, variable amounts of time, but they eventually terminate. And, we know that, maybe, I don't know if he tried it, but, can you evolve a cellular automata for more than 700 generations in this manner? So there's a lifetime to this process. In the tree of life, there's also sometimes a timeline to lineages. Certain lineages die off, and I don't think that's been very well studied. But in any case, he talks about this, and of course, undecidability links into the Turing machines and the ability to halt. And so he also gets into that a little bit later on. So you can actually boil this down to an even more minimal model. And so, again, we're thinking about computational rules and this sort of computational universality, meaning that I can take this and ask questions about other systems. So if I find these dynamics in this minimal model of life, it should inform things like machine learning. But in this case, we want an even more minimal model to boil down what we're finding in the model to something else. And so this is something he built called the mutation graph. So it gives us a link between these mutations as they're introduced and the fitnesses, and it gives us sort of this ability to extract a set of rules that allow us to find the best manner of sort of adaptive evolution. So, we might go back and ask, like, what does this mean for biology? Not in the minimal sense, but in the broader sense. And so how do biological organisms manage to be the way they are with all their complex and seemingly clever solution to such a wide range of challenges? So are we just dealing with natural selection, or is there something else going on? And so this is where he gets into biology and how, it's not about sort of this cognitive decision making, but it's just kind of a set of random processes. And so, we have to think about not only these random processes, but the way he views it is that they're these randomly chosen programs and they have a behavior. And those behaviors are really the basis of what we're talking about. So, he thinks very much in terms of programs and their behavior. And so this is maybe a point of contention for biology, because biologists don't necessarily think of things as programs. We think about like, gene expression programs, but those are sort of analogies. And so, there's something here that's sort of an assumption about the underlying biology, but it's interesting. So one thing he talks about here, he talks a little bit about the complexity community and how, people have been using genetic algorithms to try to solve different cellular automata rules, or, evolving different types of optimization procedures. And so this has a lot of links to evolutionary computation as well as biology. So it may be, that we're looking at a minimal model of evolutionary computation more than biology. He also brings up Greg Chaitin's work on metabiology. Greg Chaitin published a book, I think it was in 2009, on metabiology, where he wrote sort of a proof of Darwinian evolution. And so it's kind of an interesting book. I remember reviewing it in a blog post at the time. And so it was really, it was very reminiscent of this work, but also some of the work they do in Avita, which is an artificial life platform, basically taking evolution and boiling it down to some of these concepts that we see here. That we see these discrete states, we see natural selection, and we see evolutionary dynamics. And so it's a really interesting kind of linkage there as well. And of course, Greg Chaitin's done a lot of things with algorithmic information theory and so forth. And so that's all. This is a very long article. If you're interested, it's on Stephen Wolfram's blog.